I think now we can start with the morphology session. And our first speaker is Angelika Manhart from UCL. And Angelika, please feel free to share your screen anytime.
Yes. Can you see my screen?
Perfectly, yes.
Yep. And you can hear me all right?
Yes, that too.
Okay, brilliant. Should I start?
Yes.
Okay, thanks a lot. Yeah, so I'm very happy to be able to join you. So I'm joining from London. So for some of you, that's probably the other side of the Atlantic. And I'm sure on your side of the Atlantic, if you're there, you're looking forward to a lunch break. Whereas in the UK, it's more closer to an afternoon tea break, I would say. So today I want to talk about nuclear growth dynamics. And in general, so this falls in the category of understanding scaling laws in biology. And this has been of interest for a long time. So in the 1930s, so the person called Max Kleiber discovered a scaling law, how the body mass of organisms relates to their metabolic rate, so to their energy consumption. And when he plotted that on a log scale for a large variety of organisms, so from incredibly small to very large organisms, he found a scaling exponent of 3/4. And in general, scaling laws are an observation of some data. But of course, if they're observed over large ranges of scales or many organisms, they often point to some fundamental concepts that are behind it. But per se, they don't tell you something about the mechanism, but they pose good questions about what mechanisms could be behind a certain observation. So I want to talk about nuclear scaling. So I'm talking about cells, and cells, or all higher cells, have a nucleus where the DNA is stored and where it's the first step in protein production. And it has been observed that in general, for most cells that have one nucleus, which is your standard cell if you want, that the nuclear size scales with cell size. So if you have a larger cell, you have a proportionally larger nucleus. So a number of people have done some very interesting experiments with multi-nucleated cells. And so there's a good review by Levy and Heald, where they reviewed some experiments that were done with fungal cells. So they have several nuclei. And you can see, for example, here on the left, that if they have a cell with 4 nuclei, and then they split it, but split the nuclei unevenly, you can see that the one that has sort of a larger amount of cytoplasm to itself will grow larger. Right? And a similar phenomenon you can see in this cartoon on the right, where again, the nucleus in the middle grows larger. And so that indicates, or is a strong indication, that it's something in the cytoplasm, so in the material outside of the nucleus, but inside of the cell, that actually causes the nucleus to grow. And there have been, of course, a number of experiments to suggest different components that are responsible for allowing the nucleus to grow to a certain size. One project that I worked on together with Alex Mogilna at NYU, Mary Bayliss and Stephanie Windner at Sloan Kettering, was that we looked at another type of multinucleated cells that are maybe more relevant to us, which is muscle cells. They also have multiple nuclei, as opposed to your standard cell that just has one. And here we worked with fly cells. So each of those blue rectangles is a fly cell in a developing fly muscle. And here you can, and the nuclei are the yellow circles. And you can see, for example, from this picture, here you have 2 fly muscle cells that are roughly the same size, but they have a different number of nuclei. And you can already see that the cell that has more nuclei has smaller nuclei than the cell that has fewer nuclei. So you again see this kind of scaling relationship between cell size and now total nuclear size. And we quantified that for this project. And you can see that if we look at cell area compared to total nuclear area, and here we talk about area because the cells are very flat. So area is an okay proxy for volume. You can see a very, very strong correlation between cell area and total nuclear area. Of course, this is sort of tells you something about global regulation. But as we saw before with the experiments with the nuclei in the fungal cells, we also want to understand something about local regulation. And for that, we examined a hypothesis that is called Myonuclear domain hypothesis. So this is the idea that in a muscle cell, that a nucleus, and they're called myonuclei because they're in muscle cells, sort of caters for or supplies its surrounding cytoplasm. And so we had to define some type of domain. There's of course no set sort of borders in this cell, but you can define domains by using a Voronoi tessellation. So in this sketch below, each dot would represent a nucleus, the rectangle would represent the cell, and the different colors would be the different areas that you get when you do a Voronoi tessellation. And indeed, when we do that with the data, we find that those nuclei that have a larger Voronoi area around them indeed grow smaller. So this is a local regulation at the local level. Now, of course, if you want to have a mechanistic model that explains why nuclei would grow larger, a relatively intuitive model is that we assume there's some signal that is created in the cytoplasm, or that is in the cytoplasm, that diffuses around. And when it hits a nucleus, it becomes absorbed. And that is the signal or the component that allows the nucleus to grow. So we can simulate this, this scenario. And you can see for this example cell, which is a cartoon of a real experimental measurement, you can see that we have numbered the nuclei 1 to 14. And then we have in green the real measured nuclear area, and the black stars is the simulated nuclear area according to this idea that you have a diffusible signal that is being absorbed and then causes the nucleus to grow. And you can see that we get very good agreement, and we can, of course, we did this for a larger data set, but I think this already demonstrates quite nicely what we've seen for the fungal cells before, that a nucleus that has more area around it will grow larger. What we did not have for this project was dynamic data. So time dynamic data that would show us how the nucleus grows in time. Because for this project, we worked with fly larvae, so they were killed when we imaged them. So another interesting question is, of course, dynamically, what is the time scale of the nuclear growth? And for this, I want to talk about a different project, so a more recent project, together with my PhD student, Vivian Leach, with Jay Gatlin and Alan Lindsay. And so what Jay's group does is they do very nice in vitro experiments. And so they work not with flies, but with frogs. And what they can do is they can extract, they use extract of frog eggs. And then they create sort of cell mimicking droplets. So they have a machine or a contraption where they have the extract of the cells, and then they use oil, and then they create these droplets. And each droplet then cannot interact with its surrounding, but each droplet initially does not have a nucleus, but the nucleus is formed de novo, so starts to grow. And because we have, this is in vitro data, we can image it, and we can get very nice time course data about how the nucleus forms from nothing to having a certain size. And so the experimental data looks like this. So we have now a time resolved data. So the different lines always represent the nuclear radius, and each line is the average over a number of different droplets of similar size. So the droplets, I will sometimes say droplet size and sometimes cell size, but I mean, they should mean roughly the same thing. So you can see, for example, for smaller droplet sizes, the nuclei grow and then reach what looks like a steady state. And then for larger droplet sizes, they are still growing at the end of the experiment. And so this we wanted to understand using a model and also testing different hypotheses that have been suggested biologically. And because the system is so simple, right? You have these spherical droplets and a single nucleus inside. We can analyze our models in a more detailed way than we can do with more complicated geometries. And so the model still has the same basic ideas. So it models the dynamics of some nuclear growth factor or system signal U that depends on space and time. And we assume everything is radially symmetric, which is an approximation, but probably reasonably justified. So the domain on which the signal or the nuclear growth factor lives is outside the nucleus, but inside the cell, and the cell has a radius, a fixed radius of RC. and the nucleus has a dynamically changing radius RT. And so the signal diffuses, it cannot leave the cell, and it is absorbed with a certain absorption constant kappa that I will say more about in a moment at the nuclear membrane. And so how does this absorption work? So biologically, everything that all medium or large-sized molecules have to be imported into the nucleus by what are called nuclear pore complexes. So these are kind of controlled gates where molecules can be imported that have the right labeling. Yeah? And it is reasonable to assume that the absorption constant, so how quickly the nuclear growth factor is absorbed into the cell, into the nucleus, will depend on the nuclear pore complexes. Now, so far, I have not told you how the nucleus grows, so how R changes dynamically in time. And so if you focus on the right-hand side, what this says is that the change in time is proportional to, so we have 4 pi r squared, so that's the surface area of a nucleus of radius r, multiplied by the signal concentration at the nuclear surface, and then multiplied by the absorption constant kappa and some proportionality constant alpha. And the set of R is the measure of nuclear size that is affected by the signal. So we don't know per se if that is the nuclear, if that is the radius, the surface area of the nucleus, or the volume of the nucleus. And that of course depends on what is causing the nucleus to grow. So for example, lamins, so they are involved at the nuclear membrane, so they play a role at the nuclear membrane. If it's lamins that are imported into the nucleus, then we would, for example, expect maybe a surface scaling. If it is something that interacts, that has also been suggested and measured with the histones, so another component of the nucleus, it might be volume scaling. But we wanted to test the different hypotheses and see what fits best with the data. And then the second set of hypotheses we wanted to test is what happens with the density of nuclear pore complexes. So as the nucleus grows, does the density of the surface density of nuclear pore complexes stay constant because they have to be incorporated into the nucleus? Or is it in fact set initially proportional to the size of the cell and stays constant, so that the number stays constant, but then the density of course goes down because they become diluted by growth, right, as the nucleus grows? And we wanted to test both hypotheses, and because we have this dynamic, time dynamic data, we can do that. So this is the basic model, and it's not very complicated, but it's still sort of a certain computational effort to solve it, also because it is a free boundary problem, and so you have to put some thoughts into how you discretize your PDE. We then realized that at least for small droplets, that we might be able to go to a certain parameter regime, where a non-dimensional parameter is small. And this parameter, it depends a little bit on which hypothesis you're looking at, but it's basically to do with cell size compared to diffusion constant of the nuclear growth factor. So you can imagine that if the cell is small or diffusion is large, then the signal concentration, the nuclear growth concentration, factor concentration will be relatively constant in the cell, and then we're in a well-mixed regime. So if we do that, if we take this limit, the situation becomes much easier. So for each of our hypotheses, we just have one nonlinear ODE. Right? So that's much, much simpler than having a coupled PDE with a free boundary coupled to an ODE that is nonlinear. So now we only have one nonlinear ODE that we can, of course, analyze relatively straightforwardly or also simulate very quickly. Okay? So we did some parameter fitting, looking at those 6 different hypotheses. So different scaling, radial scaling, surface scaling, volume scaling, and constant nuclear pore complex density versus constant nuclear pore complex number. And for each of those, we determined the best parameters. So we have a 2-parameter family that we have to look at and determine the error to the data that we have. And so we found the minimal error so it was 6.3% per measurement, but it's not so important. And we found some parameters that gave us a best fit. And of course, you can also look at how sensitively the error depends on the parameters. Right? And so this would indicate, which is consistent with the biological literature, that it is indeed volume scaling, so that the volume, the nuclear volume, the ratio of nuclear volume to cell volume is constant if it's fully equilibrated. But we also get information about something to do with the time dynamics, and that suggests that nuclear pore complex density is in fact constant through the growth of the nucleus. So that means that as it grows, more NPCs have to be incorporated. So if we inspect this visually, we have here the experimental data and the best fit result. We see that for small droplets, we have a very good fit. Right, we see very nicely that that looks very similar. For medium-sized droplets, it still looks quite convincing, but for very large droplets, we see that experimentally they grow slower than what the model would suggest. And you could say, well, maybe that's because here your scaling assumption is violated. But in fact, even if we looked at an approximate spatial model, even the full model, we only get a slightly better fit, but it does not fully explain why the nuclei in the very large droplets or cells grow so slowly. So that, I think, it's a model limitation. We did one other test. So the model predicts that if you normalize your measurements by the cell radius, that then all the measurements of nuclear growth per time should fall on this line. And again, we see that for small or medium-sized droplets, that fits very well, but that the very large droplets actually grow slower than predicted. A second thing I wanted to show you to compare to experimental data is still in the frog. But now what we have is, this is early frog development. So you initially have one very large egg cell, so it's just one cell. And then there's a series of rapid divisions. And at each division, the cells divide. So you have an exponential growth in number of cells. And by division 12, you have more than 4,000 cells. But the total size of the sum of all the cell sizes stays the same. And so in general, the model suggests if the nuclei can always equilibrate, that the ratio between nuclear volume and cell volume would always be the same. Right? But what was observed experimentally is that initially, this ratio of nuclear to cytoplasmic volume is actually very small and then grows. So this is sort of time or number of divisions grows and then only becomes constant later on. Right? So you could say, well, is that in contradiction to the model? And the answer, it is not, because the model, there is a time scale involved in that, right? So the nuclei don't always have time to equilibrate. If they were always equilibrated, then they would always reach a constant ratio. But if you sort of restart the process every time the cell divides, then the nuclei will take longer in the bigger cells to equilibrate. And that means that the ratio will initially, so especially for sort of shorter division times, so say, for example, this yellow dashed line, right? In larger cells, which is what you have at the beginning, they will take longer to equilibrate and will not reach the full ratio. But then as the cells get smaller and smaller, the nuclei can equilibrate faster because growth happens faster. And then they will reach this constant ratio that is predicted at equilibrium by the model. So those are two examples where a relatively simple model can explain different sets of experimental observations. But of course, there's lots of open questions, and we are relatively sure that there will be more things we have to add to this quite sort of minimal model. So for example, there's probably more than one nuclear growth factor that could dominate at different times of development. There could be other transport mechanisms than diffusion, for example, microtubules, where the signal is transported. Maybe the dynamics of the nuclear pore complexes could be more involved than we think, and maybe that explains why the very large nuclei don't grow as quick as the model would suggest. Right? And there might also be effects of nuclear export. So these are all things that we've only started to look at recently, and that will probably lead to a refinement of this model. And I'm going to stop here, and I'm happy to take any questions.
So thanks a lot, Angelika, for your interesting talk.
So, are there any questions? I have one more. I don't know if there is someone. So, would you mind going back to your slide with your diffusion equation model?
Sure.
Yeah. So, yes, perfect. Exactly this one. I was wondering, if I remember correctly, when you evolve a sphere or a circle, even in the plane, using, for example, the mean curvature flow, you when you go to the equation of the curvature, that has a cubic term. Which somehow is plays some sort of role of a reaction term, which I believe goes against the diffusion. So
do you mean the fact that sort of the material inside as it grows, right? That it might deform the material inside of the cytoplasm?
Well,
is that what you're referring to?
I didn't take that perspective, but it could be the biological version of what I'm talking about.
Yeah, if it's that, so you're right that if you assume that the cytoplasm inside is compressible, right? So then it would get deformed or squashed as the nucleus grows, and then you would have an additional transport term.
A different type of term then it's not there.
Yes.
Yeah, I understand the drift term. I'm thinking about a nonlinear term, something like u cube, with a sign which is opposite with respect to the diffusion.
Okay, and that would, because I mean the equation is posed inside of the cytoplasm, right? It does not live on the membrane, so it's not it is.
Yes.
I'm not entirely sure if I understand.
I'm saying instead of using directly the diffusive point of view, think about evolving the U through mean curvature flow. That automatically will give the an extra term in the equation for the curvature.
Okay.
Which is a nonlinear term, and it's some sort of reaction term. Now, the difficulty there is that there are solutions which blow up, but I believe that there is a short-time existence anyway. And that might control the slow growth that you see in the data. I don't know if it is true, but it might be a perspective.
Maybe we should talk about this
on Slack chat
some more, because I think I don't completely understand where the term could come from.
Yes.
But I also don't want to keep people from having a deserved lunch break.
Sure. Are there other questions?
I had a quick one. Well, several, but I'll keep it to one quick one. And that was if the goal here, or if these cells tend to maintain sort of a constant ratio of the nuclear volume to the cytoplasmic volume, there are a couple ways to achieve that, right? I mean, you can do it through many smaller nuclei versus a few larger nuclei. And I'm kind of wondering which strategy is better, and if you see differences in those strategies across taxa.
So, I mean, across different species, I don't really know. So, I mean, I've worked more with the fly muscle cells than I've never done anything on the fungal cells. So what we noticed for the fly muscle cells is that sort of there is a rough correlation between the number of nuclei and the size of the cell. So as if the cell sort of says, okay, so I roughly estimate, let's say for this cell size that I want to have, because I'm a muscle cell, so I have to work that hard, I need 10 nuclei, but plus minus 5. And then if I don't get it right, I just adjust the size of the nucleus accordingly. So if I have more nuclei than I roughly estimated, then I just make them smaller. And if I have, did I say it now the right way? You know what I mean, right? If you have fewer, then you make it larger. So maybe sort of adjusting the size here is a way, sort of an extra level control mechanism. So if you're first you say, okay, you can't have just one nucleus here, because the transport of moving products around, the distances would become very large.
Right.
And so there's probably sort of an intermediate number of nuclei that would be good, something between not too many and not too few. And then you have a local or a sort of an extra level of control to fine tune the size of the nucleus, which then has to do with sort of supplying the muscle cell with whatever it needs. Could that make sense?
Yeah, absolutely. Thanks.
Other questions?
Hi.
Thomas?
Hi. So I have a question about some assumptions that you presented, especially the one that actually signal comes from the cytoplasm. Yes, that cytoplasm somehow communicates with nucleus and about size. I really like the hypothesis about nuclear pore complex, and I was wondering, can you support your model by some evidence coming from mutations in export and import factors? Would they change the size of the nucleus?
So for import, it has been very clear that if you mess with the import, you can affect nuclear size. So these people know. For export, there's different evidence, and that might be because it depends on the organism, or it might be depends on the cell state or cell type. So for export, there's both experimental evidence that if you mess with export, it does not affect nuclear size, or that it does. So that's just, it seems that there is not an answer across all species, as far as I understand the literature. Yeah.
Okay, thank you.
So that's why we did not initially include it, but it will depend on the results of our experimental collaborators. If in their experimental system it turns out that export does play a role, then we will include it.
But import mutants will have increased size of the nucleus?
So if you hinder import, I think what usually is observed, and I would have to double check if that is consistent in the literature, is that the nuclei are smaller. But I don't know if that statement is true across different organisms, because I would have to check. But it's clear that nuclear pore complexes are important for nuclear growth. So that if you hinder their ability to import substances, then you do get smaller nuclei.
It's very interesting and striking, because we always think that nucleus controls everything, and that would actually be very, very contradictory to that.
Yeah, well, I mean, but once they are formed, of course, then the whole protein production starts at the nucleus, but they need to form initially, and every time the cell divides, they need to form a new, right? So there is, I think that depends probably on who controls what, depends a lot of also on the time that you look at in the development of the cell or the organism.
So another follow-up, maybe some idea is that probably the factor that is imported should be heavily conserved across evolution.
I mean,
do you have some idea what this could be? Yes.
So I mean, of course, this is I presented a simpler story than what is probably true. Different things have been observed. So lamins, which are components of the nuclear, of the membrane between the nucleus and the cytoplasm, they have been suggested as such a nuclear growth factor. Then some molecules that interact with histones, some nucleoplasmins, have been suggested as a nuclear growth factor. So as I understand it, that there's probably not just one component that controls fully the size of the nucleus. It also wouldn't make sense, because of course the nucleus needs many components to form and to function. So I do think that lamins, for example, I think they've been observed to play a role in nuclear size scaling in many different organisms. But to say, there's just one nuclear growth factor is certainly not giving justice to the complexity of the system.
So that would be simply shortage of building material.
I mean, that has often been suggested, that sort of the nuclear growth factor is just some material that builds the nucleus, but it could be more complicated than that.
Great, thank you so much.
Thanks for your questions.
Well, thanks a lot for interesting questions, and of course, many thanks for Angelika for her very nice presentation and for coming to our workshop. And with this, I think we conclude the morning session, but not the morphology session, because we'll start again at 1:45 with our keynote speaker, Professor Miriam Zeldic. And in the meantime, I would strongly recommend to visit our virtual environments, in particular Gather Town, and the Slack chat for further details about the next talks. So thanks a lot to everybody for the interesting morning section.
Thank you.
And we resume in 1 hour and 45 minutes.